Hi, welcome back to your next video lecture. This one over viral pathogenesis, or how viruses affect cells and how they cause disease in the body. I particularly like this quote because I think it's fitting, since a lot of the pathology and pathogenesis we see with viruses is actually due to our own immune system and not necessarily to the virus itself. All right, so here's our case study for the day. And what I want you guys to think about as we move through the next few classes where we're talking about viral diseases is how are you going to distinguish between a bacterial or viral illness? So in this case study, we have a 5-year-old child who presents to her primary care physician with a sore throat, dry cough, headache, and conjunctivitis. So the physician passes it off as just a cold until he examines her mouth and notices tiny white lesions with red border on her cheek membranes. And upon seeing these, he is sure of his diagnosis. So you can see these white spots right here, along with their red borders on the inside of her cheek. So here's our question. How are you going to tell the difference between a viral and a bacterial disease with similar presentations? We've talked about many bacteria that can cause sore throat, they can cause a dry cough, headache can be a similar symptom. So how will you notice the differences between these two? And the key is that there's often something very small, like white spots inside the mouth, that will tell you what disease it is and whether it's bacterial or viral. Okay, so today we're going to learn about cytopathic effect and be able to explain the cytopathic effect, or the effect on cells, and its importance in viral replication and disease. Generally describe the techniques that we use to measure virulence. Explain how both host factors and viral factors contribute to pathogenesis. Describe several ways in which viruses may interfere with or counteract immune responses, similar to how we did for bacteria. And then understand, just at really basic levels, how viruses manipulate the cell and drive oncogenesis. We will talk more about oncogenesis when we get to TBL3, where you will learn about human papillomavirus. Before we jump in and start talking about the different things that viruses and cells do that cause disease, we need to spend just a little bit of time talking about the different patterns of infection we can see with viruses. So each of these graphs is showing us time and the amount of virus being made in the body over that time period. So A here shows us just a really acute infection. You get sick, you have some period of disease, and you recover, and the virus is cleared from your body. There's no more virus there, right? So this is your common cold. This is when you get the flu and you get better. The second one is a latent infection. So these always begin with an acute phase, and then the amount of virus in the body disappears. The virus is dormant. We don't get new viral replication until sometimes later, and it could be weeks, it could be months, it could be years before reactivation occurs. And when reactivation occurs, we now see the production of virus, and that's usually associated with symptoms. So this would be like a herpes virus infection, where you get cold sores. You have your initial infection, it disappears for a while, a cold sore appears again a few months later, it disappears, a new one appears, and so on and so forth. C is showing us an acute infection followed by a chronic infection. So again, we have our acute phase where a person's going to be ill, they're making a lot of virus in their body, and then it tanks out. But rather than being cleared, we see a low level of viral replication that can persist for years or even the life of the patient. This would be something we might see with hepatitis B or hepatitis C. D shows us a totally different scenario where, again, we have an acute infection, and this goes down kind of like a chronic one to what we're calling a persistent infection here. So the virus is staying around in the body. It's being made at very low levels for a very, very long time. And then at some point, there's some sort of switch or trigger that happens where we go from this low set point level of viral replication, and the amount of virus recoverable from the body starts increasing again. And as that increases, symptom severity increases too. So the prime example of one that does this, that has an acute infection, followed by a set point persistent infection, and then ramping back up, is HIV. And then the last one is the slow chronic infection. So a person initially gets infected, they maybe don't experience any symptoms, and it begins to slowly increase over time, and our person gets more viral production and perhaps worsening of symptoms over time. Right, so those are the 5 different patterns of infection. When we talk about an acute infection, a persistent infection, what we're referring back to is the virulence of a particular virus. So virulence is defined as the ability of a pathogen, a virus or a bacteria, to cause disease in a host. So there are viruses that can be very virulent. They can cause a lot of disease in the host, very bad disease. And those that are avirulent. Avirulent meaning they don't cause symptoms, they don't cause disease. And this can vary based on a number of factors, even within the same host species. Now we can actually quantitate virulence. We can put numbers to it. We can measure the virus titer, or the amount of virus being made in an animal, in a human, in a particular type of cell in culture. We can measure the time to death in our test animals or in a naturally occurring outbreak. We can look at the time to the appearance of signs and symptoms. So how long is the incubation period for a particular disease? And how severe are those symptoms? So in animals, we can, in some animals, measure fever. We can measure weight loss, which is a sign of illness in mice and other rodents. And we can measure physiological changes. So we can look for the appearance of something like lesions. We can measure the number and type of immune cells, and so on and so forth. Cytokine production, muscle tone, anything that could be related to our viral infection. But an issue here, again, is that many of these signs and symptoms are caused by our own immune responses. So it can be tricky to tease out which of these are caused by the virus itself and which are caused by the immune system. Take fever, for example. That's a result of inflammation and immune system activation. So we can see it in viruses, we can see it in bacteria, and we can even see it under times of stress. So if we're going to measure the virulence of a particular virus in vitro in cell culture, we need to remember a few key things first. So in the last video, we went over the different outcomes of a virus infection. So there was an abortive infection, a persistent infection, that could be chronic or latent, a lytic infection, and an oncogenic infection. So these you should review on your own and make sure you remember what these are before continuing on. Now, when we infect cells in a petri dish, one of the main things we're going to look for is cytopathogenicity, which is a big word that means changes to the cell because of viral infection. We can also state it like is written here, the ability of a virus to cause degenerative changes in cells or cause cell death. These changes we refer to as cytopathic effects, and we shorten that to CPE. So we'll look at a cell and we'll determine the type and the amount of CPE after infection. Now, attenuated viruses have lost the ability to cause cytopathic effect. They can still replicate in those cells, but they don't cause CPE. So what might attenuated viruses that replicate but don't cause adverse effects be used for? If you're thinking those might be useful in vaccines, you're absolutely right. There are several viral vaccines that are actually live attenuated viruses. They cause a really robust immune response, but they don't cause disease in the person receiving the vaccine. Okay, so what does CPE look like? It's this kind of weird amorphous word. There are a lot of different ways that this can happen, and one is that we can see CPE in the nucleus. So we can see physical changes to the nucleus just looking at our cells in a regular old microscope. Inclusion bodies, or these large aggregates within the nucleus, is one. We can see that in the picture here. So adenovirus, which causes the common cold, it causes pink eye, a few other types of diseases, replicates in the nucleus. And if you look at these cells right here that I'm circling, these are normal nuclei. So the pink with the dark purple spots, those are normal nuclei. This arrow, you can see the nucleus is smaller and it's really dark. That's our adenovirus infected cell. Those inclusion bodies are actually full of newly assembled adenovirus particles just waiting to lyse the cell and be released into the extracellular space. Some other changes you can see are a thickening of the nucleus, swelling of the nucleus so that it takes up almost the full cell body, changes into the nucleolus, or margination or condensation of chromatin, all of which can be visible in just a regular microscope. We can also see a lot of changes to the cytoplasm or the organelles of our infected cells, depending again on where that virus replicates. Inclusion bodies, again, is one of the big things that we can see. We can also see vacuoles or stranding within the cytoplasm, so the formation of like really thick strands of proteins in the cytoplasm. Here we're looking at an electron micrograph of a cross-section through the endoplasmic reticulum of a cell that's been infected with dengue virus, which is a flavivirus. And the arrow is pointing to these inclusion bodies here, which are basically little viral replication factories. So those are full of viral proteins and viral genomes that are starting to assemble, and then they're going to move through the ER and Golgi before they're released at the cell surface. So the nucleus, the cytoplasm, or other organelles can see a cytopathic effect. We can also see very easily changes to our cell membrane. So the two easiest ones to see here are that our cells can round up, which we can see in these pictures up the top. So this top left quadrant of the picture are normal uninfected cells. They're flat, they are attaching to each other, they're kind of triangular or teardrop shape. After being infected, we can see here at 5.5 hours, some of these cells are starting to round up. So all of these little, they almost look like bubbles that you can see here that I'm circling with the pointer. These are cells that have rounded up and are no longer flat and attached to the dish. As this infection continues, here at 8 hours, you can see almost all the cells are rounded up. They're still down at the bottom, but by 24 hours, they've pulled away completely from the dish, and now they're just floating around in the liquid. So cells will round up and they'll eventually lose their adherence and come off the dish. This picture at the bottom shows us the formation of syncytia. And a syncytia is essentially a multinucleated giant cell. So over here I'm circling a regular cell. It's flat. You can see the margin of the membrane, and you can see the nucleus with its nucleoli on the inside. This giant, big, ugly looking thing that you see in the middle, this is a syncytia. And if we look at it, you can see it's huge. The margins of the membrane are huge, and packed inside it are many, many nuclei. I haven't counted. I would guess there's maybe 20 or 30 nuclei in this one cell. Now, this is not a viable cell. It can survive for a while, but eventually it's going to die. This whole big syncytia is going to round up and pull off of the plate all at once. Some viruses, like respiratory syncytial virus, are really, really good at causing this. And essentially, it's a fusion of the cell membranes that are all there really close together. And as those membranes fuse, we go from a single nucleated cell to a multinucleated cell. And then, of course, we can just look for cell lysis. Are our cells no longer there? Have they been burst open? Then they have been lysed. So here we can see, to some degree, cell rounding and cell lysis. Here's our normal cells. These are a different type of cells. They're very long and thin. After a few hours, they have started to lyse. So we have the loss of cells here, where there used to be cells filling up our whole field of view. And down here we see that there are very few cells left. What cells are left tend to be rounded up and stuck to our petri dish. So you may be wondering, like, well, why is it important to know that there are changes to the nucleus, or that there are changes to the endoplasmic reticulum or the cytoplasm? And that's because the more we can understand how a virus affects an individual cell, the better we can understand how it replicates, the better we can work to find effective treatments that will prevent cell death, that will prevent syncytia formation, that will prevent chromatin condensation, and thus alleviate symptoms to our host. But what about viruses that don't show any detectable CPE? What if we infect cells with this virus and the cells still look completely normal? Can we still detect them? And the answer is yes. Even though we may not see changes to a cell in culture, we can still detect our viruses. So we can do this in vitro using a plaque assay, which I've decided not to talk about here because it's just not that pertinent that you understand how a plaque assay works. But we can also measure virulence in vivo. So perhaps there's no change to cells in a petri dish, but we will see change to an animal's behavior, an animal's inflammation, if we measure virulence in vivo. So I have two different graphs here. One really easy way to measure virulence is to simply do a survival curve. So in this little graph, we've got some mice that have been infected with virus type 1 and virus type 2. We can see that infection with virus type 1, all of the mice live. With virus type 2, they are all dead by 10 days post-infection. So that's a really easy measure of virulence. Do the animals live or do they not live? We can also look at the tissues that may be affected upon infection and look more where in the body is virulence happening. So this graph is showing us the relative neurovirulence of various types of flaviviruses, and we're looking at what region of the CNS is most affected by these particular viruses. So we see with Japanese encephalitis virus, the cerebellum and the brainstem are mostly affected, and the spinal cord not as much. We see that this is less on all counts with yellow fever. We see differences with West Nile, where it's the brainstem that's mostly affected, and dengue is not really neurovirulent at all. So we don't really see any neurovirulence in any part of the brain. So even though these are closely related viruses, they have very different patterns of virulence on the CNS. But virulence is relative. So what's virulent in one animal may not be virulent in another. What's virulent in one human may not be virulent in another. So that kind of brings up these two questions. And I've posted these on the discussion board on World Class, where you can kind of give your thoughts here. What host factors might influence the severity of disease, and what viral factors might influence the severity of disease? So for this first one, it can include things like we've already talked about, like age, immune status, any underlying health conditions, but it could be things at the very cellular level, like do they have a particular mutation that makes the virus not replicate as well or increases viral replication. As for viral factors, this could include things like immediate early or early enzymes that change the cell cycle and drive S phase of the cell cycle, or that turn off a particular type of immune response. And those could influence the pathogenicity and the virulence and the severity of symptoms as well. So we're going to talk about some examples of each of these, starting with host factors. So this one in particular is my favorite example of a host factor that's required for viral replication and disease manifestation. So this is a host microRNA called microRNA 122 or miR-122. It's only made in the liver. So that's why hepatitis C is liver trophic, because that's where microRNA 122 is made. And this microRNA is required for hepatitis C replication. So if we treat patients with a drug that prevents microRNA 122 from associating with the hepatitis C genome, that reduces viral replication in humans. So these two diagrams are showing us how this works. This whole structure here is the hepatitis C virus RNA genome. These structures are part of the tertiary structure of the 5' untranslated region of the genome, upstream of all of the genes. So miR-122 binds to and associates with the viral RNA, with the viral genome, and this actually facilitates replication of our viral RNA to make more genome copies. So when we have miR-122 produced, we get a lot of virus replication, we get a lot of cell death and pathology in the human liver. When we treat with something like a anti-microRNA, so it's a little RNA that binds to the microRNA and prevents it from interacting with the 5' UTR, we do not get efficient viral replication. So without miR-122 binding, we don't get a good amount of RNA genome replication. We don't get the production of new viruses. And it's really effective and really efficient. And I think it's pretty cool. So that's a really great example of host factors that are required for viral replication and that are highly implicated in the disease state. But more often, we focus on viral factors or viral virulence genes. Just like when we talked about bacteria, we talked about bacterial virulence factors. So if we were working in a lab trying to identify viral genes that are important for virulence, for the causation of disease, the easiest way to go about this is to make some mutant viruses. So here on the top, we have what we call our wild-type virus. That means our normal virus, like we would see that normally causes disease. When we grow it in cell culture, it grows a lot. We have a whole lot of virus that comes out. And then when we inject that virus into mice, it replicates in the mice and it causes disease. And for our scenario here, we get neurovirulence. So the virus replicates in the brain and it causes issues with the brain. As we create mutations, we may generate a mutation that just limits reproduction of the virus. So now it doesn't reproduce, it doesn't grow very effectively. So we don't get robust growth in culture. It's a really weakened virus. Because it doesn't grow well, when we put it into animals, it's still not going to grow well, even worse than it does in culture. And we call this an attenuated virus. Okay, it's the same strain of virus, but we've mutated it now so that it doesn't replicate very well. At this point, we've just created a mutant that has a defect in reproduction. It doesn't mean that it's really less virulent, it's just that it doesn't grow to the same amount as our wild type. In this bottom scenario, we've created a mutation in a gene that is required for virulence, but not required for reproduction. So when we grow it in culture, we still get a whole lot of growth in culture. But when we put it into an animal, it does not reproduce very well because that gene doesn't prevent it from growing in culture, but does prevent it from growing in mice. So again, we call this an attenuated virus because it doesn't grow very well. Okay. There are a number of types of viral virulence genes. By and large, viruses don't produce toxins like bacteria do. They don't really produce exotoxins that they secrete out because they're intracellular pathogens. So most of their virulence genes are focused on altering the cell that they're currently in and perhaps altering its state. We do consider genes that are involved in viral replication to be virulence factors, because without these, there will be no virulence. Viruses can also produce genes that allow for greater invasiveness, so that can allow them to spread from cell to cell very easily, like formation of syncytia. Tropism, again, so we can have tissue-specific tropism or species-specific tropism. And these are related to virulence. So we only see neurovirulence in viruses that are capable of infecting some sort of CNS cells or other neurons throughout the body. And of course, under this umbrella of virulence genes are the viral genes that are going to modify host defenses. And like with bacteria, this is a really big goal of many viruses. As we saw in the diagram on the last slide, some genes are not always required for growth in vitro, but they may be required for growth in the host. So if we look back, I'm going to flip back really quickly. If we look back here, this gene allowed, a mutation in this gene still allowed the virus to grow really well in culture, but it was attenuated in the mouse itself. So the question I have for you is why? Why would a mutation preclude replication in an animal, but not in the cell? And the quick answer is that must be a gene that's involved in modifying host defenses in some way, like downregulating interferon responses, something to do with immune evasion. And in cell culture, in vitro, there is no immune system. It's just one type of cells. So you still grow just fine, but then you put it into an animal that has an immune system, and all of a sudden that virus can't grow. And of course, genes that enable the virus to spread throughout the host, that allow it to spread from the initial site of infection, maybe a mosquito bite, and get into the endothelial cells throughout the body, like we see with dengue. And the very last one are those oncogenic gene products, the viral genes that drive oncogenesis and cancer formation in our cells. And those are usually genes that drive the cell into the S phase of the cell cycle. We're going to talk about those a little bit at the very end. Okay. There are a number of larger scale host factors and host defenses that are important both in fighting off an infection and also in causing pathogenesis. And of course, the first is the immune status of your host. An immunocompromised host is always going to be more susceptible to infection, but sometimes, like in the case of the H5N1 bird flu epidemic, having a robust immune status can be a detriment. The genetic background of our host can also be really important. You may have heard of the CCR5 delta 32 mutation. A very, very small percentage of the population has this mutation, and it actually makes them resistant to infection with HIV. Age, like with bacterial pneumonia, is really important. As a person ages, they're more likely to experience shingles, which is caused by varicella zoster virus, to experience various cancers caused by Epstein-Barr virus. And that, again, it's also a matter of immune status and the weakening of the immune system as people age. Nutrition is important. We always know that people who have inadequate nutrition are more susceptible to all types of infection. Hormones also play a pretty important role, especially during pregnancy. Those hormonal changes alter the immune status, and so you can be more susceptible to various types of infections at different times of your life based on the hormones your body is producing. Pregnancy is one of those times that you can get infected really easily because your immune system is decreased. Now, host defenses, right? You guys have learned about these in immunology. We have a lot of chemokines, the production of interferons, and those specific cells like our NKs, CTLs, and the production of neutralizing antibody, which all work to fight off viral infections. So just really briefly, the antiviral response of cells usually is in response to the recognition of double-stranded RNA. So a lot of viruses generate double-stranded RNA as a byproduct of RNA replication or messenger RNA production. And this recognition turns on the interferon response. And you guys have probably learned about 2' to 5' oligosynthetase, which again, its goal is to turn on ribonucleases that will degrade messenger RNA. These can degrade host and viral messenger RNA with the goal of preventing viral protein synthesis. If we prevent viral protein synthesis, we can't make new virus. We prevent the spread of the virus throughout the body. Additionally, interferon production can turn on our protein kinases, again in response to double-stranded RNA. And what a lot of these protein kinases do is they inhibit EIF2 alpha, or elongation initiation factor 2 alpha, which is important for protein synthesis. Again, if we don't make viral proteins, we don't make viruses, our host doesn't get sick. So our body tries to shut everything down by the interferon pathway as soon as a virus is recognized. Unfortunately, a lot of viruses are really good at preventing interferon signaling or decreasing it, usually by the degradation of STAT1 and STAT2 or preventing the dimerization of STAT1 and STAT2. But there are a number of other viral gene products that alter host immune responses, and not just the interferon response. So viruses can make products that can interact with BCL2 to either prevent or induce apoptosis of our infected cell. So if you're a virus, it'd be really great to prevent apoptosis, because then you can keep using that cell as a factory for more viruses. The longer the cell stays alive, the more viruses you can make. Viruses can also produce what we call virokines or viroreceptors. So virokines would be similar to chemokines, so they might bring in an immune response that may be beneficial for viral replication and viral spread. Viroreceptors can be viral mock receptors that can be put on the surface of cells that cytokines may bind to, but then there's no further signaling pathway, or they can be released from the cell and help to neutralize cytokines that would normally interact with the cell. So they prevent activation of the antiviral responses. Viruses, interestingly enough, can also bind to complement binding proteins. It's been shown that mumps and some other enveloped viruses actually accumulate factor H in their envelope because the complement-mediated killing can work on some enveloped viruses as well. Another really interesting thing is that a lot of viruses downregulate MHC class 1 and class 2. They can cause the internalization of MHC, so the cells bring it back out or back into the cell. So any MHC that's expressed on the outside of the cell then gets internalized and degraded, so they're not signaling to the world around them that they've been infected. You guys learned in immunology about how herpes viruses can interfere with the loading of peptides into MHC by interfering with TAP. And there are other ways that viruses can prevent presentation of viral antigens to the immune system. And they can also modify our pattern recognition receptors like toll-like receptors. Again, these can be internalized off the cell surface. Those that are internal TLRs can be degraded or otherwise inactivated so that the host just does not recognize that there's a virus there at all. This chart, again, you never need to memorize a chart. I'm just giving you some examples of how viruses can do all these things that I've just told you about. So I'm only going to point out a couple here. So humoral immunity and antibody production. Herpes simplex virus and cytomegalovirus, which is also a herpes virus, can encode their own FC receptor. So this will bind to the FC region of antibodies, and this blocks effector functions of antibodies bound to infected cells. So that Fc receptor is bound, and it prevents the antibody from recognizing the infected cell. Down here on our inhibition of inflammatory responses, again, let's talk about Epstein-Barr virus here, EBV, that causes mono a lot. They can produce, or they can inhibit the expression of adhesin molecules like ICAM1, which you've probably learned about are important for attachment of lymphocytes to infected cells. So by inhibiting the production of these on infected cells, our lymphocytes cannot attach to those cells and they can't clear them. Blocking of antigen presentation. Again, you guys learned about herpes viruses doing this in immunology. Here's herpes simplex right here with the inhibition of peptide transport via TAP. And in that way, we don't have antigen presentation by MHC class 1. And some viruses can actually cause immunosuppression of the host, not just preventing immune responses specific to the virus, but general immunosuppression. So Epstein-Barr virus, EBV, encodes a homolog of IL-10, and it forces cells to produce this IL-10 homolog. By doing that, it inhibits Th1 responses, reduces interferon gamma production, and generally causes immunosuppression. Right, and that's something that we see in chronic EBV and in mononucleosis. So viruses can interact with the immune response in order to prevent a response from recognizing infected cells, but the immune system can also cause damage to your body. And again, I've said this before, I'll say it again, a lot of the pathology we see during viral infection is caused by our immune system and not necessarily by viral replication itself. So our CD8 T cells can be pretty important for immunopathology. And this makes sense, right? Because a CD8 T cell response is going to be the primary response in clearing an intracellular pathogen. So for infection with viruses like Coxsackie, Hep B, and HIV-1. A lot of the pathology we see here is due to CD8 T cell mediated killing of cells that have been infected with these viruses, so lung cells, liver cells, and our CD4 T cells. On the other end, we can have disease mediated by CD4 T cells. Our Th1 cells can be important. We see this with measles. We're going to talk about that in a second. And again, with herpes simplex viruses, we'll talk about that one too. We can have Th2 mediated CD4 T cell pathology. This happens in respiratory syncytial virus. And then B cell mediated immunopathology. This one we see with dengue virus. This we call antibody-dependent enhancement, where those antibodies to one type of dengue virus actually facilitate infection with a different type of dengue virus. And it seems that we can also see that they facilitate infection with Zika virus as well. So first, to kind of diagram out how these CD8 T cells are involved in disease, we're looking at this case at LCMV, which is lymphocytic choriomeningitis virus. It's a mouse virus, so we're looking at the percent of mice that survive an LCMV infection when they either produce perforin or cannot produce perforin. They lack the gene for perforin. So over time, at day 8, we start to lose some of our mice that produce perforin, and by day 10, all of those mice have died. However, in mice that cannot produce perforin, they all survive despite infection with LCMV. So this tells us it's not the virus itself that's killing the animal, it's the production of perforin. So what's happening is actually destruction of liver. So our graph on the right here is showing us perforin production again. And in those that do not produce perforin, we don't see liver enzymes being released into the serum. We do see an increase in liver enzyme activity in our perforin-positive mice. So we're causing tissue destruction of the liver and death to our animals, and that's mediated by our CD8 T cells. With our CD4 T cells, we can see in general with those, right, they don't directly mediate cell killing, but they do increase inflammation. So our CD4 T cell mediated diseases are going to be associated with higher inflammation. And this inflammation is going to recruit and activate non-specific effector cells. So neutrophils, mononuclear cells, but not specific CD8 T cells. As these neutrophils and mononuclear cells come to the site of inflammation, they release proteases, free radicals, and other cytokines. And this inflammation can cause a variety of types of damage. In herpes virus, this can cause what's called a herpes stromal keratitis. So this is when an eye is infected with herpes virus, and those CD4-Th1 cells come to the eye. So the virus replicates in the epithelium of the eye. The immune response as it comes in attacks the stromal cells. And you can see that there's thickening here in the eye, that it's cloudy, it's been blocked over. This is a pretty common cause of blindness in developing countries. Some of those toll-like receptors are also really important in initiating antiviral responses and in causing inflammation. And they can, again, be associated with the disease we see. So you've probably heard of West Nile virus. It causes encephalitis. It can be fatal in many cases, and even when it's not fatal, it takes a very long time to recover from. What we know is that mice lacking TLR3 are resistant to West Nile virus infection, and they also don't produce large amounts of cytokines. We know that in mice that can produce TNF-alpha, that TNF-alpha compromises the blood-brain barrier, leading to encephalitis and swelling. So in our image here, we're looking at wild-type mice that have TLR3 and can produce TNF-alpha, and we're looking at those that lack TLR3. So day 0, this is a normal mouse brain. It looks nice and healthy. What they've done is they've infected mice, then they've injected them with a dye called Evans blue. Now, if vasodilation has occurred and we get leakage of blood, or we get encephalitis, inflammation, leakage of blood into the brain, we'll see an influx of blue dye. So you can see that here. In our wild-type mice at day 3 post-infection, we have a lot of Evans blue accumulating in the brain, telling us that the blood-brain barrier has been compromised. In our TLR3 knockout mice, we don't see any Evans blue dye. They are still doing okay. Out to 5 days, our West Nile infected mice still have some accumulation of blue. It's not as bad as day 3, and it's only at this point in our TLR3 mice that we start to see any of that occur. But these mice tend to recover just fine, whereas these mice tend to die or need to be sacked. All right, so back to our case study at the top of the lecture. We had our 5-year-old child who presented to her PCP with a sore throat, dry cough, headache, conjunctivitis, and white spots on the oral mucosa. And these white spots are called Koplik spots. Now, these Koplik spots are a hallmark of infection with measles, whose name is rubeola virus. The rest of the symptoms, the cough, the headache, conjunctivitis, sore throat, these are pretty nonspecific. It could be a number of viruses causing this. It could be a number of bacteria causing this. So only these Koplik spots this early on told our physician that this child had measles. Now, if she had not come to her PCP at this point, within a few days, the child will likely have developed the rash that we think of when we think of measles, but we can identify early on in the prodromal phase based on these Koplik spots. So it's often something really small that can help us distinguish between like sore throat, dry cough. Maybe she's got, the beginnings of pneumonia, or meningitis because of the headache, and something like measles. So measles is really good at immunosuppression. One of the strategies of immunosuppression is to prevent a reaction from occurring. So instead of getting around the immune response, hiding from the immune response, just don't let an immune response occur. So here we have on the left side a virus that's interacting with TLR4. When this virus interacts, our cell starts producing IL-12, and this turns on CD4 T cells, and we're generating a Th1 response. So we're generating the response that's going to lead to CD8 T cell activation and the killing of infected cells. This is what we, as a human, want to happen so that we can be nice and healthy, clear out the virus, and recover just fine. What measles does is it still interacts with TLR4. It still gets recognized, but it prevents the expression and secretion of IL-12. So we don't get IL-12 made. We don't generate a Th1 response. Instead, we generate a Th2 response that's going to favor antibody production. Antibody production can have an important role in clearing viruses, but antibodies alone are not going to do it. You need those CD8 T cells to kill off the infected cells. So measles just shuts down our CD8 T cell response, pushes everything toward antibody production. And this doesn't just have effects during the initial infection. What we've actually found is that post-measles infection, children will still have immunosuppression for up to years, and they don't generate good CD8 T cell responses for some reason. So a few more examples, right? Rubeola virus, that's measles. It can infect monocytes, dendritic cells, thymic epithelial cells, as well as epithelial cells of the skin, where we get the red rash from. And as it infects these and carries out its effects, we get reduced T cell activation and enhanced infections, both enhanced measles infection, but also enhanced bacterial infections, even after the measles has been cleared. Rubella virus, this is another part of the MMR vaccine. It's measles, mumps, and rubella. These viruses infect lymphoid cells, and they dampen the immune response so that a person can have persistent rubella infection and can have the virus replicating in them for a long time. As that happens, your immune system becomes tolerant of the virus. It no longer really tries to fight it off. And if that happens in a pregnant woman, it can lead to infection of the fetus. And we'll talk about the fatal and terrible consequences of fetal rubella syndrome later on in the semester. And of course, we all know about HIV. It does cause AIDS, acquired immune deficiency syndrome. HIV infects CD4 T cells and it infects monocytes. And as it inhibits our CD4 T cell responses, as it inhibits a variety of other immune systems, our HIV patient, as they develop AIDS itself is characterized by a number of opportunistic infections, such as fungal infections that we don't normally see in an otherwise healthy individual, and the development of rare cancers. All right, and the final thing on pathogenicity, again, that we're just going to touch on is oncogenic transformation. So transformation in bacteria means the acquisition of new genes. Transformation in a eukaryotic cell means we have transformed it into an immortal cell, a cancerous cell. for all cancers, but particularly when we're talking about viruses, there has to be a two-pronged approach here. We have to have both of these two things happen in order for the development of a sustainable cancer. And the first is that we need to do something with an oncogene. So our virus either needs to produce its own oncogene or alter a cellular oncogene. So there are several viruses that produce MYC, which is an oncogene. If it comes from the virus, we call it vMYC, which stands for viral MYC, because virologists are not creative in naming things. So they can produce their own MYC, which is going to drive cellular replication, drive the S phase of the cell cycle, or they can simply turn on the pre-existing cellular MYC, which we call cMYC. Either way, you get the same result where you're telling the cell, divide, 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 divide. Another really popular pathway for viruses to activate is the RAS pathway, which again does the same thing. It tells the cells it's time to divide uncontrollably, and we're losing one of those checkpoints that prevents the development of cancer. But this alone is not enough to make a sustainable cancer. Sure, you can drive the cell cycle, which for many viruses is necessary for effective viral production. But eventually, those tumor suppressor proteins will lead to apoptosis of our infected cell. So the second part of this two-pronged approach is that oncogenic viruses need to shut down tumor suppressors, inhibit tumor suppressors from doing their job. And all viruses that cause cancer target p53. They can do one of these things, two of these things, or all of them. A very common thing is p53 degradation. So the virus will utilize host pathways to ubiquitinate p53 and target it for degradation. Others will simply sequester p53, so maybe take it out of the nucleus and put it in the cytoplasm where it cannot recognize double-stranded DNA breaks, where it can't turn on the cell death pathways. And then still others will actually interfere with gene expression and downregulate p53 expression. What we often see is a combination. So a virus might downregulate p53 expression while also degrading any p53 that's currently in the cell so that we completely deprive cells of p53. So we have to have some combination of both of these happen in order to drive oncogenesis and to drive cancer formation due to a virus. So there are DNA viruses that can cause cancer, and there are some RNA viruses that can cause cancer. We've all heard of papillomaviruses, right? HPV. Their acute phase is warts, genital warts. But they do cause cervical carcinoma, along with they can cause anal carcinomas, oral cancers of various types. Hepatitis B virus, if you remember, that's the really weird one that has the partially double-stranded, partially single-stranded DNA genome with the covalently bound RNA. I believe it's a Baltimore class 7 virus. This one causes hepatocellular carcinoma. Epstein-Barr virus, which is a herpes virus, can cause a number of different types of cancer. So it can cause Burkitt's lymphoma, nasopharyngeal carcinoma, and B-cell lymphoproliferative diseases. Merkel cell polyomavirus is, I believe it's the newest one discovered that can cause cancers in humans. It causes this really rare skin cancer called Merkel cell cancer. There have only been a few cases of this so far. We don't really know where all in the world it's occurring. And then for our RNA viruses, we have hepatitis C, which causes liver cancer. Human T-cell leukemia viruses, there's two of those, HTLV1 and 2. These cause adult T-cell leukemias, but we mostly see them in Japan and in the West Indies. We don't see too much of that in the United States. And then we can have a combination of an HIV positive individual who's also infected with human herpes virus 8, which is also known as Kaposi's sarcoma-associated herpes virus. And when you have this unique combination of infections, your patient can wind up with Kaposi's sarcoma. We don't see Kaposi's sarcoma in really any other individuals except those that are infected with HIV. Okay, guys, that's all for today. Much shorter than the last one. Next up, the next lecture I'll be posting is about antiviral drugs and the drugs we use, the limited arsenal of drugs that we use to fight off viral infections.